Welcome back to BinaryBio. If you've been following our Rosalind series, you've already mastered calculating GC content and finding a shared motif. But today, we are moving from simply reading sequences to understanding how the cell edits them. We're tackling the RNA splicing problem. In the real world, a gene isn't just one long set of instructions. It's a rough draft that requires a heavy cut and paste job before it can build a protein. In eukaryotic cells, genes are split into two types of segments: exons and introns. Exons are expressed, they contain the actual protein coding information. Introns are intervening, they are non coding gaps that must be removed. The cellular machine that handles this is the spliceosome. It identifies the introns, snips them out, and glues the exons back together to create mature mRNA. If this process fails by even a single letter, the entire resulting protein could be non functional or even toxic. Once the introns are gone, the cell has to translate that mRNA into a protein. It does this by reading the sequence in three-letter words called codons. There are 64 possible codons, but only 20 amino acids. This means multiple codons can code for the same amino acid, a concept called redundancy. For example, AUG is the start codon, telling the cell where to begin, while others like UAA or UAG act as stop signs. In this problem, we have to simulate this exact process, stripping the introns and then translating the remaining exons, three letters at a time. Why does this matter in 2026? Because alternative splicing allows one gene to create multiple different proteins by mixing and matching different exons. This is how humans achieve such incredible complexity without needing millions of extra genes. More importantly, splicing errors are a massive target for modern medicine. Diseases like spinal muscular atrophy, SMA, and many cancers are caused by splicing gone wrong. By solving this problem computationally, we build models that can predict how a mutation might create a fake splice site. We are now even designing splice switching drugs that act as digital tape, covering up bad splice sites to fix genetic diseases at the RNA level. In our Python solution, we treat this as a multi step string manipulation. We parse the DNA and the list of introns. We then loop through the introns and use dot replace to remove them from the main string. Finally, we iterate through the remaining exons in steps of 3 to map each codon to its amino acid. When we analyze our solution, we need to consider how it performs as the DNA sequences get longer. The time complexity of our splicing phase uses dot replace for each intron. If n is the length of the main DNA and m is the number of introns, the complexity is roughly O of m times n. While efficient for Rosalind datasets, this could become a bottleneck for entire chromosomes. Our space complexity has a complexity of O of n because we create new string copies each time we remove an intron. While our custom script is great for learning the logic, in an industry setting, we wouldn't rewrite these dictionaries from scratch. The standard production ready approach is to use established libraries like BioPython. Industry professionals use seek objects because they are optimized for memory, handle ambiguous bases like n, and offer high performance methods for transcription and translation that are far more robust than a simple Python dictionary. Using industry standard libraries ensures your code is maintainable, biologically accurate, and ready for integration into larger genomic pipelines. Splicing is the biological edit that makes human complexity possible. If you found this video helpful, hit the like button and subscribe to BinaryBio. We're here to help you navigate the intersection of code and biology. Thanks for watching, and I'll see you in the next one.